Hi, I'm Adarza, and I'm here with another lecture. Well, another lecture in the Explicit Memory Storage lecture series. Again, we are at section 3. And, well, I told you that in section 3 I'm just going to introduce brain microcircuits, and in the last video of all of these lectures I'm going to give you an introduction to hippocampal microcircuits. But in this lecture, in previous lectures I introduced microcircuits. I talked about the historical background on microcircuits. I also talked about the microcircuit studies. And now in this lecture, I'm going to talk about cell assemblies, because they are important parts of microcircuits. And finally, in the next part of this lecture, I'm going to talk about the dynamic aspects of microcircuits. And in a separate lecture, I'm just going to go and do that introduction on hippocampal microcircuits. Okay? So just in this lecture, I'm going to just talk about cell assemblies and the dynamic aspects of microcircuits. Okay. Let's go to the cell assemblies first. Well, first thing first, let's define cell assemblies. Well, as I understood cell assemblies are just a group of neurons that fire at the same time. Well, this is a very informal kind of definition. But the formal definition of cell assemblies, according to one of the papers I studied for this lecture, is that, well, a single cell assembly is an anatomically dispersed, sometimes anatomically dispersed, set of neurons among which excitatory connections and recurrent excitatory connections particularly are strengthened or potentiated. And that's why they actually, when one of them fires, the whole set of neurons which participate in a single cell assembly are going to be co-activated and are going to fire at the same time, okay? So, well, that's a definition of cell assembly, and now let's see how they work, how a single cell assembly is activated. For instance, first of all, we can have a sensory input, which is in this figure represented by these red arrows. And then this sensory input can drive some of the neurons participating in a single cell assembly, these colored neurons, not these white neurons, but those colored neurons. This sensory input can drive some of the neurons in a single cell assembly to fire. And when one or two or a few of them actually fire, because there is a strengthened recurrent connection among the members of a single cell assembly, represented by these black arrows, these strengthened recurrent connections, which they are going to stabilize and keep on the firing of the cell assembly. They are going to temporarily and transiently stabilize the firing of a cell assembly through mutual excitation. Okay, so they're all the members of a single cell assembly are connected to each other. The neurons of a single cell assembly are connected to each other through mutual excitatory connections, and once one of them fires, or a few of them fire, the others are going to fire and stabilize the firing of that cell assembly. But sometimes, the excitability of a single cell assembly is going to fade gradually. But at this moment, because there are some inter-assembly connections, okay, represented in this figure by these blue arrows, there are some inter-assembly connections, and these inter-assembly connections are going to subsequently activate a new cell assembly or a new set of cell assemblies, okay? And by doing that, they actually create a phase sequence. Well, the word phase is not related to time or timing here. Basically, the definition of a phase sequence is just a phase sequence is a chain of cell assemblies, each one is in which each one is triggered or is activated by the last one. Okay, for example, what you see here is actually a phase sequence, because this cell assembly is triggered or activated by this one, and this one is triggered by this one. And the mere fact that these cell assemblies can be activated by other cell assemblies actually tells us that we don't need that sensory input. We don't always need that sensory input, because sometimes a single cell assembly can be activated by internal factors, including other cell assemblies. Okay, and finally, that phase sequence, that chain of activated cell assemblies, is going to evolve based on the principles of network dynamics and the neuronal dynamics of these neurons in a single in cell assemblies. And basically, that the evolution of that phase sequence is what is hypothesized as the cellular substrate, or the substrate of internal cognitive processes. This is what scientists think is the substrate of our cognitive processes, of our thoughts, imaginations, memories, and all that thing, all those cognitive faculties and systems or processes in our minds. The evolution, the changes, and the the dynamics of that phase sequence, or a chain of cell assemblies in a single microsecond. I think, I want to ask you this question. If you are watching this video, I hope you are watching this video, you 5 professors, and I want to ask you a question because, I think, according to what I studied, I think a single cell assembly is part of a bigger structure, or is part of a bigger microcircuit, actually. So we have microcircuits, and then we have cell assemblies within a microcircuit. If I'm wrong, please correct me, okay? Because I think that this is true, because we have a big microcircuit comprised of, again, I explained in the previous lectures that a single microcircuit is comprised of principal neurons and interneurons, and then we have that complicated structure, and within the microcircuit we have cell assemblies. So if I'm wrong, please correct me. Thank you. So this is how a single cell assembly is activated, and it can activate other cell assemblies in order to form a phase sequence. And well, a phase sequence is then evolved, is going to actually evolve, and the evolution of that is very important for our cognitive processes. Okay, so the question is, excuse me, it's a little bit warm here. So the question is, okay, how scientists detect a cell assembly, or how they know that a group of cells form a single cell assembly. And this is one of the methods they actually do. For instance, this is a 2-photon calcium imaging of a slice from V1 area, the visual area 1. Okay, and this is a contour plot. And in a contour plot, you can see it depicts or demonstrates the contours of individual cells, and this is a contour plot related to the same graph here, or image here. And these are the contours of around 754 neurons, okay? And then what scientists do, is that they actually use a raster plot, okay, to detect those neurons that are active at the same time or fire at the same time. So this is a raster plot. It's a fantastic way to demonstrate a lot of different features of those neurons that fire together. So in a raster plot, each row represents a neuron, and you can see we have approximately 700 neurons, because this is the raster plot for these two images. So we have, each row represents a single neuron, and each one of these marks represents a calcium transient. Sometimes each mark represents an action potential or firing. In this case, because sometimes the calcium transient or an increase in intercellular calcium concentration may be considered as firing of an action potential in a single cell. So that's why we actually relate these two together. Relate the increase in intercellular calcium concentration to action potentials or to firing. Okay, so anyway, these marks, each one of these marks is a single calcium transient. And you can see that some of these neurons, we can see some vertical lines that indicate that some of these neurons fire at the same time. And actually, I put some boxes or some borders around those vertical lines so that you can see them better. For instance, there's this vertical line which represents that all of these neurons at this particular time all of them fire, and they're all active. And there are other vertical lines, okay, representing those neurons that are active at the same time. And then there is this histograph related to this raster plot, which actually tells us the percentage of those cells that are active at the same time, or at the same moment. And you can see, for instance, at this particular time, a large percentage, a great percentage of those cells are activated. Okay, and it's related to this part, this vertical line. And scientists neuroscientists know that those cells that are activated at the same time, or they fire at the same time together in a synchronized way, they are probably parts of a single cell assembly. Okay, and this is one of the methods that scientists use to detect those cell assemblies. About the spatial organization of cell assemblies, well, I told you in the definition of cell assemblies that they're anatomically dispersed ones, but sometimes they're not completely dispersed. Sometimes they can form clusters. So these are all contour plots, and these red colored cells or neurons are neurons that form a single cell assembly, or are part of a single cell assembly. You can see that sometimes they form a cluster. Sometimes they have some dispersed patterns. Okay, they're dispersed within different layers and columns. Sometimes they're all form a horizontal line within a single layer, or they can form a vertical line within a single column. Okay, but of course they fire together, but that they can have different spatial organization. That doesn't mean that they need to be close to each other. Okay, they can be very far from each other, but still fire together. Okay, so that was about the spatial organization of cell assemblies. And now there's a movie, and here I want to show you why I think single cell assemblies are parts of microcircuits. So here we are going to see a model. I think it's a computational model of a synthetic microcircuit, or an in silico microcircuit, which is composed of only principal neurons or pyramidal neurons and some inhibitory neurons. Let's watch it together. So we have, I told you that in microcircuits we have pyramidal neurons or principal neurons and interneurons, which are primarily inhibitory. And these pyramidal neurons are those principal neurons, and we have inhibitory neurons, and then we add more of these two types of neurons to get a whole microcircuit, or a small microcircuit. It's not a very big microcircuit. But it is still complicated, of course. And then what happens here is very wonderful. At each time, some of these, not all, but some of these neurons actually fire, okay? And they fire at the same time together, okay? So some of these neurons are activated at each time. They fire at each particular time, and I think they are part of a single cell assembly. Okay, and that's why I think cell assemblies are parts of a bigger structure called microcircuits. So you can see that at each time, different neurons fire together. Okay, so they can be part of a single cell assembly. This is from Allen Institute for Brain Science. It's a very popular institute, and is doing a lot of big projects in neuroscience, of course. And this movie is from the Blue Brain Project by Professor Henry Markram, that I introduced in the previous lecture. And here you can see the neurons' membrane potential is actually indicated by the range of colors. You can see from blue, which is minus 80 millivolts, to almost a little bit white, minus 10 millivolts, and the time here. And you can see again that at each time some of these neurons, this is a whole microcircuit, okay, and some of these, and sometimes, as you saw, we have a flood of, we have a a big action potential activating all, which is a result, a big signal, which is a result of the activation of all a very large number of neurons. But you know, this is the same concept. This demonstrates the same concept as the previous video in the previous slide. You know, at each time, some neurons fire at the same time together, and those neurons are part of a single cell assembly. Okay, so that was all I wanted to talk about cell assemblies, and now I want to talk about the dynamic aspects of microcircuits. Excuse me. Okay. Well, I told you that those canonical microcircuits that I introduced in the second lecture, the previous lecture, has two problems. First of all, they're not very successful in demonstrating the structural complexity of real microcircuits. And they have another problem, and that is they cannot actually demonstrate the dynamic aspects of microcircuits, because a lot of aspects of microcircuits, including the connectivity between synapses, are dynamic. The connectivity between neurons, or synapses are dynamic, okay? And they change over time, they're plastic actually. And so now I want to tell you that how scientists are trying to understand the dynamics of microcircuits or neural networks. And I want to start by a theory by one of the great and giant neuroscientists, called the theory of neuronal group selection. Professor Gerald Edelman is the man I want to talk about his theories in this part of the lecture. Well, Professor Gerald Edelman was a biologist who won the Nobel Prize in Physiology or Medicine for discovering the structure of antibodies. But later in his life, he contributed a lot to neuroscience by several different theories and ideas, and one of those theories is the theory of neural Darwinism. Well, the basic concept behind neural Darwinism is that Gerald Edelman, as well as some other scientists and philosophers, and of course me, I'm not a scientist, of course I'm a student in science, but I also agree with Professor Edelman. Well, Professor Edelman, in his theory of neural Darwinism, he believes that the brain is not a Turing machine. It is actually a Darwinian machine. And so I'm sure that you are all familiar with the Turing machine. The Turing machine is a computational machine which is basically the mother of all of our electronic computers nowadays. And it was developed or invented by Alan Turing, one of the fathers of modern computer science. And that machine, I told you it's a computational machine, and it actually solves some problems based on the algorithms that we give the machine, and it works based on ones and zeros, okay? The building blocks of the language of our modern computers. But we know that the brain is not a Turing machine. The brain doesn't work with ones and zeros. The brain is a computational machine. It is a computer, but the brain is an organic one. Is a biological computer. And as Dr. Gerald Edelman says, it's a Darwinian machine. It means that it's a biological entity, and like many other biological things, it evolves over time. And even Daniel Dennett, who's a philosopher of mind, believes that, well, I told you what he believes. He believes that brain is a computational machine, but it is not like our modern computers nowadays or cell phones. It is a biological machine. It's a biological or an organic computer. It means that its principles of computation are not similar to the principles of Alan Turing's machine. Okay, so and in a book explaining his theory, Neural Darwinism, Dr. Gerald Edelman also introduces the theory of neuronal group selection. And this theory tells us that the the process by which neurons are selected for individual cell assemblies or ensembles. Ensemble is another name, I think, for cell assemblies, or microcircuits. The process by which the neurons are selected for microcircuits are dynamic, and they change during both development and based on the behavior that we have, okay, or the experiences that we have. So let's see what this theory is all about. So I told you that we, according to Professor Gerald Edelman, we have two types of selection. We have a developmental selection, and of course, as its name suggests, developmental selection happens during development. And you can see that these neurons at time 1 are not, some of them are connected to each other, but some of them are not. And they are actually going to connect to each other and form new connections, and some of the connections are going to be deleted by natural processes during the development. For example, cell division and cell death, the elimination or extension of neuronal processes, and some molecular mechanisms, including the growth factor signaling transduction pathways. And so you can see that at time 2, some of these neurons, they form new connections, and some of the older connections are deleted. And this kind of selection, that yields the primary repertoire of our microcircuits that we are born with. Or I love to call that primary repertoire the default networks that we are all born with, okay? And then we have another type of selection called experiential selection, which, again, the name suggests what it means. The name actually is obvious. It's a type of selection, it's a type of neuronal group selection during experiments, or a type of selection that happens because of experience, because of our interactions with our environment. For instance, we have a stimuli here, and at time 1, the stimuli, okay, and then the stimuli causes some changes in the strength of synapses. It may strengthen some of the synapses and it may weaken some of the synapses. And at time 2, you can see that some of these lines representing the synapses or connections between these dots or neurons are bolded, it means, or thickened. It means that those connections are strengthened. And then we have these dashed lines. These dashed lines represent those weakened connections. And this kind of selection or experiential selection yields our secondary repertoire of microcircuits, which are much more personal, I think, than the primary repertoire. Because the stimuli we are exposed, they may vary greatly, okay? From person to person, from individual to individual. And then we have also reentrant mapping, which is basically, it says that even the connection between maps of the brain, which are, I told you in the very first lecture on microcircuits, that maps are one level higher than networks, okay, or microcircuits. And maps are basically, I think, there are areas in the brain. Even the connectivity or the connections between different maps of the brain, different areas of the brain are dynamic, and they may change over time based on stimuli and based on our experiences and behavior, okay, and interactions with the environment. So Dr. Gerald Edelman knew at that time that, and proposed a theory or a model for this feature of microcircuits, that the feature that they are dynamic and they change over time. The neuronal connections between in a single microcircuit actually change over time. They're plastic. They may get strengthened or weakened. Okay. This is my favorite part. I want to talk about the neural networks and their interaction with gene regulatory networks. So, the dynamic aspects of microcircuits are not just limited to the dynamics of synaptic contacts between neurons that comprise a single microcircuit. Sometimes those dynamic aspects are also related, can also be related to the molecular pathways, the molecular networks within individual neurons, okay? And that tremendously increases the level of complexity in microcircuit studies. But I do love this kind of topic a lot, and let me explain that to you. Well, we have gene regulatory networks. We all know what neural networks is. Of course, you know what neural networks are. Of course, you know what gene regulatory networks are, but again, I'm going to explain them. Well, gene regulatory networks are complicated networks of proteins, mostly transcriptional factors and co-activators and related proteins, that they orchestrate or regulate the expression level of individual proteins within the cell and in the cell. And according to experts in that field, in the field of genetics, we have two types of gene regulatory networks. We have developmental ones and behavioral gene regulatory networks. Well, again, the name can tell us what they are and what they are about. The developmental gene regulatory networks, they control the gene expression or the changes in gene expression during the development. But what's interesting is that we have some gene regulatory networks that they control the expression of some particular proteins as a result of behavior, as a result of our interactions with the environment. And those are called behavioral gene regulatory networks. And scientists think that behavioral gene regulatory networks are the molecular substrates, of behavior or cognition. Okay? And so now let's see how that interaction happens between neural networks and gene regulatory networks. You know, some signals may be carried by neural networks, and those signals can sometimes activate a single gene regulatory network in one neuron. And then that gene regulatory network may activate another gene regulatory network in another neuron by neuroendocrine signaling. And then the behavioral gene regulatory networks and sometimes also the developmental gene regulatory networks, they affect the neural network and they feed back to the neural networks and they affect the neural networks. So let's see how, let me give you some examples. For instance, the temporal kinetics of neural or neuronal firing, or basically the firing pattern of a neuron, may activate or may interact with some gene regulatory networks in a single neuron. It's wonderful. I mean, the firing pattern or the temporal kinetics of neuronal firing of some particular neurons may act on gene regulatory networks in the cell, in that particular neuron. And that is the example of how neuronal activity or the activity of neural networks affect or interact with gene regulatory networks. In another example, gene regulatory networks actually affect the activity of neuronal networks. For instance, the expression of a particular protein, okay, which is controlled by a gene regulatory network, may have some impacts on the firing properties of a single neuron. Okay, it may change the firing property of a single neuron. So this is an example of how a gene regulatory network, particularly a behavioral gene regulatory network, may impact the activity of a neuronal network. And usually the behavioral gene regulatory networks, they influence the activity of neural networks by setting the production level or the expression level of neurotransmitter receptors. Okay? For example, if a single cell produces more neurotransmitter receptors, or here it is called neuroreceptors, it means that the sensitivity of that particular cell is going to be increased. The sensitivity to chemical signals that it receives. It increased, like the amplification or the insertion of new AMPA receptors in the postsynaptic membrane, okay, as we saw as I explained it in the expression of line term potentiation, okay, or the expression level of ion channels, or for example, some gene regulatory networks, particularly behavioral gene regulatory networks, can control the expression of some proteins which are important for axonal outgrowth or dendritic arborization. Okay, so basically these behavioral GRNs or gene regulatory networks, they can control several aspects of the physio and chemical. They can actually control physiochemical aspects of neural networks. Okay. And sometimes these behavioral GRNs can act together with developmental GRNs, and they can cause some morphological changes because of those developmental gene regulatory networks. They can change the neuronal wiring, or the connectivity of between these neural networks, within these neural networks, and they can cause some morphological or structural or physical changes in individual neural networks. It is wonderful that, you know, how molecular activity in individual cells may have some impacts on the whole neural network. It's amazing. So these two are two of the papers behind the examples I gave you in the previous slide, the examples of the interactions between gene regulatory networks and neural networks. For instance, in this paper, published in Scientific Reports in Nature, it is an example of how neural networks affect gene regulatory networks. For example, it says, the title of this is Gene Networks Activated by Specific Patterns of Action Potentials in Dorsal Root Ganglion Neurons. So a specific firing pattern of a specific cell type may have some impacts on a gene regulatory network and may activate some gene networks. And in this work, they did a lot of bioinformatic studies and techniques. And in another paper published in Science, it is an example of how gene regulatory networks, affect neural networks. For instance, there is a tuning of fast spiking interneuron properties by an activity-dependent transcriptional switch. So we have a transcriptional switch, which is, its expression is actually controlled by a gene regulatory network, and that gene regulatory network can affect, or that protein can affect, the firing properties of a cell type. So thank you so much for watching this video. That was it. I just wanted to talk about the dynamic aspects of microcircuits. And yes, that's it. So thank you so much for watching this video. I hope you can watch other videos and other lectures. And well, in the next lecture, which is going to be the last lecture, hopefully, I'm going to explain the hippocampal microcircuits. So, thank you. Have a nice day.